Hello and welcome to the webinar. Today we will talk about profiling the small RNA modifications. First, we will briefly review small RNA classes and their functional mechanisms. Then we will focus on the small RNA modifications and regulations, and how to best profile small RNA modifications. Briefly, RNAs smaller than 200 nucleotides long are small RNAs, which include microRNAs and tRNA-derived small RNAs or tsRNAs. tsRNAs include tRNA half molecules or tRNAs and further processed products as tRNA-related fragments or TRF. Small RNAs do not have sufficient open reading frames to encode proteins as mRNAs do. They are, therefore, non-coding RNAs. Non-coding RNAs longer than 200 nucleotides long are non-coding RNAs, or lincRNAs. As small RNA, microRNAs are first transcribed as primary microRNA transcript from the host gene, processed by DROSA DGCR8 process enzyme into pre-microRNA hairpin precursor, exported from the nucleus into the cytoplasm, and processed by dicer into short microRNA duplex. The guide strand is the mature microRNA loaded into AGO-containing RNA-induced silencing complex RESC to suppress the target mRNA. There are two major mechanisms of microRNA silencing, that is, repressions on translation initiation and mRNA decay. As a small RNA class, tsRNAs are not random degradation products from a full tRNA molecule. Rather, they are site-specific cleavage products with newly defined biological functions. They form a new small RNA class. When a cleavage site is within the anticodon loop by angiogenin, two fragments approximately half the size of a full RNA molecule are generated, that is, 5' and 3' tRNA halves. TRFs are the further cleavage products by dicer, angiogenin, and RNase G. Based on the regions in the parent tRNA, TRFs can be grouped into 5' TRF5, internal ITRF, 3' TRF3, and 3' tail TRF1. tsRNAs are induced under various stress conditions. For example, hypoxia, nutrient deficiency, viral infection, and heat shock, and can also be produced constitutively in the cells, such as hematopoietic stem cells. tsRNAs have lost the parent tRNA functions as amino acid carrier and mRNA decoder for protein synthesis, but have been endowed with new distinct functions in protein translation regulation to A. Act similarly as microRNAs to repress mRNA translation, B. Act on translation initiation complex to inhibit protein synthesis, C. Guide RNAs for mRNA cleavage and decay, D. Interact with RNA binding proteins, for example, YBX protein to alter the mRNA stability, E. Pause ribosome during protein translation. tRNAs also have functions unrelated to translation, for example, F. Cytochrome C binding and apoptosis, G. Tumor suppressor p53 phosphorylation, H. At a cellular level, tsRNAs are the first responder in cellular stress and are involved in cell proliferation. I. tsRNAs are most highly enriched in exosomes and become one of the most abundant small RNAs, even more so than microRNAs. J. Strikingly, tsRNAs can carry epigenetic inheritance information. Isolated tsRNAs from the sperms of high-fat diet mice can produce F1's offspring mice with diabetic phenotypes. The two main mechanisms of action by tsRNAs are transcript engagement, where the tsRNA acts as a guide RNA to recognize mRNA target by base pairing, which brings the RNA binding protein RBP, such as AGO, to act on the mRNA. This way, the tsRNA is similar to microRNA guiding AGO silencing complex to target mRNA. With this mechanism, the more the tsRNA, the more RBP action on the target mRNA, so they are positively correlated. In transcript displacement mechanism, tsRNA and the target mRNA compete for the same RBP, for example, YBX, sharing the same common binding site. With transcript displacement mechanism, the more the tsRNA, the less RBP action on the target mRNA, so they are negatively correlated. Now let's move on to the very important biological features of small RNA modifications. As shown on the top, unmodified RNAs have unmodified bases of G, A, C, U. As shown at the bottom, the bases and ribose in small RNAs can be modified by chemical groups. The most common and important modifications include OAG, N7G, M6A, M5C, and pseudouridine Si. These RNA modifications are integral to small RNA functions. For example, N7G modification can be installed by writer protein metal 1 in the quadruplex G structure in the primary microRNA, destabilizes it, and opens it up for DROSA processing. This M7G in microRNA precursor is carried over to mature LET7 microRNA, which inhibits a set of mRNAs in cell migration. RNA modifications can alter mRNA targeting specificity or activity. A exoguanine OAG modification is typically generated by intracellular reactive oxygen species ROS, making base pairing OAG with A instead of the original G with C, therefore altering the mRNA species being targeted. In case of microRNA miR-184, under oxidative stress, the OAG modified microRNA miR-184 acquires new anti-apoptotic BCL-XL and BCL-W mRNA targets, represses their translation, and promotes apoptosis. In cardiomyocytes, phenylephrine stimulation increases reactive oxygen species ROS and OAG RNA modification. In microRNA miR-1, OAG can modify the C region and base pair with A instead of C. The seventh OXO is most potent in targeting the 3' untranslated regions of cardiac genes in heart contraction, cardiac morphogenesis, hormone stimulation, cell migration, BMP signaling, and calcium ion transport. For example, the predicted 7-OXO-G site in GSK3B mRNA is in fact mappable to AGO by CLIP. The modified silencing activities of 7-OXO-G microRNA miR-1 on target mRNAs are confirmed by the reporter assays. Infusion of 7-OXO-G modified microRNA miR-1 causes cardiac hypertrophy. Transgenic overexpressions of 7U miR-1 having the same targeting specificity as 7-OXO-G miR-1 also demonstrated hypertrophic effects in mice. M5C modification in microRNAs commonly disrupt the formations of microRNA and messenger RNA duplex and leads to the loss of gene silencing activity. For example, microRNA miR181A-5P is methylated with M5C by DNA and RNA methyltransferase DMMT3A and AGO4 complex. M5C modification blocks the tumor suppressor functions of microRNA miR181A-5P and is associated with poorer prognosis of glioblastoma multiforme, GBM. Two' O methylations of the ribose at the 3' end of small RNAs protects the small RNAs from uridine addition by terminal uridinyl transferases, thus preventing the RNA entering uridination-dependent RNA decay pathways and prolong the RNA half-lives. Intriguingly, 2' O methylation prolongs plant microRNA half-life when ingested in human body, supporting an intriguing possibility that modified small RNAs derived from plant-based diet may survive long enough in the gastrointestinal tract to modulate human physiology for therapeutic use. Pseudouridine is the most common modification of small RNAs and is abundant in tRNAs and tsRNAs. Pseudouridine is installed by pseudouridine synthase 7 (PUS7) enzyme. Pseudouridine modified tsRNA in the terminal oligo G (MTOG) is very potent in suppressing protein translation, whereas unmodified tsRNAs have no such activity in the PUS7 knockout cells. The loss of pseudouridine modification impacts embryogenesis and mesoderm specification, and causes dysregulation that critically governs hematopoietic stem cell function and myelodysplastic syndromes. The way of pseudouridine inhibition of protein translation is by decoying RNA binding proteins in the protein translation pre-initiation complex. Pseudouridine modifications of the 5-terminal oligoguanosine (TOG) in tsRNAs binds and displaces poly-A binding protein PABPC1 from translation initiation complex, thus repressing the translation of capped mRNAs. Now we have seen many examples of biological functions and molecular mechanisms of small RNA modifications. To summarize in this table, small RNA modifications are known to change the targeting specificity and redirect to different mRNA targets. They can regulate the small RNA activity and efficiency, small RNA biogenesis, small RNA stability, binding to RNA binding proteins, discriminating self versus foreign in immunity, and epigenetic transgenerational inheritance. One motivation of studying small RNA modification is biomarker applications. The small RNA modification dynamics and the profiles can be indicators and biomarkers for human diseases. For example, cell damage and stress can cause tRNA cleavages to form tsRNAs, which then exposes buried M1A to antibody detection. Diabetes can alter the modification profiles of tRNAs and tsRNAs in liver, as shown by the up and down arrows. High-fat diet can install M5C and M2G in the sperm tsRNAs that can carry over the diabetic phenotype to the F1 offspring. Small RNA modifications are associated with human diseases. Together with the abundant presence of small RNAs in biofluids, small RNA modifications can be a new class of biomarkers. For example, M6A methylations of serum microRNA miR17 5P displays superior diagnostic values in pancreatic cancer in Figure A, compared with conventional gold standard carbohydrate antigen CA19-9 in Figure B, or carcinoembryonic antigen CEA in Figure C. The healthy controls colored blue and the pancreatic cancers colored red show much larger distinction and have a much lower false positive rate using the M6A modified microRNA miR17 biomarker. tsRNAs are very abundant small RNAs in biofluids, often more abundant than popular microRNAs as biomarkers. In particular, tsRNAs are heavily modified and therefore stabilized. In fact, tsRNA profiles have been demonstrated an excellent biomarker for triple-positive breast cancer cells. So how do we best profile and study small RNA modifications? A traditional way to profile RNA modification is by methylated RNA immunoprecipitation sequencing, or miRNA-seq. However, during the CDN synthesis of sequencing library construction, the reverse transcriptase stops at modification sites, so the sequencing rates lose coverage and are heavily biased towards 3' ends of the RNA. Furthermore, the multiple PCR steps used in CDN library amplification and on the sequencer flow cells in situ PCR can produce significant biases, distortion, and artifacts due to the sequence contexts and GC contents, which greatly reduce the quantification accuracy. Also, miRNA-seq does not contain modification stoichiometry information or the percentage of modification. In practice, miRNA-seq requires large amounts of input RNAs at around 100 micrograms, excluding many experiments with limiting sample amount supply. For example, small animals, small histological sites, real cell types, and precious clinical samples. Arraystar has developed small RNA modification microarrays to overcome many of the above limitations. By using specific antibodies against the modifications, a diversity of modifications, including OAG, M7G, N6A, pseudouridine, M5C, are detected. With the modified and unmodified RNAs separately labeled with two color channels, the percentage of modification or modification stoichiometry of each transcript can be quantified. One microarray simultaneously profiles multiple small RNA classes, including microRNAs, microRNA precursors, tsRNAs, that is, TIF and tRNAs. The microarrays is highly sensitive, detecting one copy of transcript per cell. The quantification accuracy is extremely high due to the faithful representations of native RNA levels by one-step labeling. Here, the 3' end of the denatured RNA is ligated to cytidine 5' phosphate labeled with silane 5 or silane 3 by T4RNA ligase in one-step ligation reaction. There are no reverse transcription, no PCR amplification to best preserve the quantification accuracy. The immunoprecipitated modified RNAs are labeled with silane 5, and the unbound unmodified RNAs are separately labeled with silane 3. So the proportions of the modified and unmodified RNAs, the modification stoichiometry, can be calculated. The microarrays are designed for human and mouse species. For human, there are more than 14,000 distinct probes covering all cataloged microRNAs, microRNA precursors, tsRNAs collected from miR-base, gtRNA-DB, TRF-DB, MIM-base, reference databases, and scientific papers. The array data are processed, annotated, and analyzed to easy-to-use Office software formats. For example, the differential M7G modification is analyzed for fold change FC, statistical significance, p-values, up or down regulation, and the stoichiometry of the percent modification changes. The microRNAs are annotated with family memberships and M7G modification motifs. The normalized signals are hierarchically clustered and presented in color-scaled heat maps for visualization. Additionally, mRNAs targeted by modified and unmodified microRNAs can be computationally predicted and displayed in Venn diagram for the common overlap and different non-overlapping targets. The modified microRNA targets are also displayed in the network graph. The differentially modified small RNAs of interest can be further studied. For example, the modified and unmodified small RNAs can have different sets of target mRNAs or targetomes. Using AGO-RIP sequencing, the small RNAs, that is microRNAs and tsRNAs, and target mRNAs are co-immunoprecipitated by AGO antibody. The precipitated RNAs are sequenced by bioinformatics analysis of the cells transfected with modified or unmodified small RNAs and the mock control cells. The small RNA targets can be identified and its targetomes analyzed. The proteins that are in complex with modified small RNA provides a wealth of functional information based on the known protein functions. To do this, biotinylated modified small RNA, for example, pseudo-urally modified tsRNA or scrambled control, are transfected into the cells. The cell lysate is poured down with streptavidin beads. The bound proteins are digested and analyzed by LC-MS, liquid chromatography, and mass spectrometry. These proteins provide the functional indication by gene ontological analysis, pathway analysis, or interaction network graphs. With the wealth of data from the small RNA modification microarray profiling, the differentially modified small RNAs can be further studied for their functions, such as transfections or infusion to study phenotypical changes in cells or animal models. The modified small RNA targets can be computationally predicted by base pairing, experimentally identified by AGO-RIP sequencing, and functionally validated by reporter assays. The protein that complex with modified small RNAs can be pulled down and identified by LC-MS, liquid chromatography, and mass spectrometry. The differentially modified small RNAs, particularly from the biofluids, can be studied as diagnostic biomarkers or with survival data for prognostic markers. The biomarker performance can be evaluated for the sensitivity and specificity by ROC curve analyses. To summarize, Arraystar small RNA modification arrays are a very practical tool for accurate and effective quantification of multiple modifications, such as OAG, N7G, M6A, pseudouridine, and M5C, in small RNA classes including microRNAs, pre-microRNAs, tsRNAs, namely TRFs and tRNAs. Thank you for your interest. Please visit our website at www.arraystar.com.